Okay, so let's get to the paper, the cringy paper that we've been working through. I had another cringy paper I had open. Where did that one go? There. Oh, that was a PDF there. Oh, well, that's fine. So before we get started, remember how we talked about how we found attractant through a big screen? So this is that original paper, which was actually published after the attractant one, even though the work for this one was done way before that. But it just happened to be that that one was easier to publish, so it got published earlier. But this is how we found attractant to begin with. So remember last time when we were talking about that paper, we found this protein. Where did we find it? We were looking for biomarkers for a very aggressive brain tumor, astrocytoma, especially the highest grade, the glioblastoma. And where did we find this protein attractant?
In CSF.
Yes, in the cerebrospinal fluid. So we were focusing on the cerebrospinal fluid for two big reasons. What were those two big reasons?
A less intrusive manner of test.
Well, slightly less intrusive than getting a biopsy.
The blood serum will have too many proteins.
Yes, so there's a lot of complexity in your blood compared to CSF. CSF is a simpler solution. And then a big one that you guys are missing right now.
It's continuous?
Well, it's continuous with the brain, which is important. Why? Does everything that goes from gets secreted from the brain? Yes, it's the blood-brain barrier. So not everything makes it into the blood to begin with, because there's that tight lock between the brain and the blood to maintain the brain in this kind of clean environment with limited exposure to toxins and to infections and all that good stuff, right? So because there's that blood-brain barrier that controls what goes in and out of it, and large molecules like attractant are very hard to get through it. They're probably never gonna make it into the bloodstream. So that is something that was part of the story, right? That's why we chose CSF, because if we were to look in the blood, many of these markers will be lost already. We wouldn't even see them. So this is actually what the gels originally looked like where we found attractant. So this is what we call a two-dimensional gel electrophoresis. These are silver stained gels. They look ugly in these pictures. They weren't as ugly in real life, I promise you. But basically, in this particular technique, we first take our proteins, we extract them from the CSF in a manner, and we solubilize them in a buffer that does not change their overall chemistry, that keeps them maintained in their tertiary structure. And so that, it maintains their typical charge that they will have inside the body, so that we can separate them simply, first of all, by their isoelectric point. So the first dimension of this electrophoresis, we separate them on this long gradient. It's like a long, thin strip that has a gradient of pH all across on it. So the ones that I was using were from pH 4 to 8. And that's, the proteins that I'm getting out then are the proteins that had isoelectric points between pH 4 and pH 8, which is majority of them. And so you first separate them by that. You then put the strip on top of, you actually treat it with certain buffers, with SDS and other agents to now linearize the proteins. And we then run them on a typical SDS page gel, like you might have seen in Western blots, right? Where you're simply separating each one of the protein by weight. And so in that second dimension, you're separating the proteins by weight so that, it's same as when we run the DNA or proteins in other ways. The bigger ones are gonna not travel as far. They'll be towards the top. The smaller ones are gonna be towards the bottom. And now you can see them separated out as spots. Instead of bands, they are individual little dots. The more protein is present, the more intense the spot, the bigger the spot. The more modified it is, the more kind of spread out the spot. So right here, this whole thing, this is albumin, because albumin, one, there's a lot of it, and two, this is all phosphorylated and bound to other proteins and very hard to get it away from other things. So you end up seeing this kind of streakiness in this big blob versus nice clean spots. The proteins that you can identify easily are the ones that are nice clean spots. These streaky ones are hard to identify sometimes. So what I did was literally take out each one of these spots. I picked them up in the gel. I put them into separate tubes. I extracted those. I actually added some trypsin. What would trypsin do? So once you have these spots in little tubes, we add trypsin to it. What would trypsin do to the proteins or the peptides in there?
Dissolve chew on the peptides.
Well, it won't dissolve them, but it will chew on them and cut them up into smaller pieces. There's no media in this one. So the trypsin will do what it normally does, that it will chew the peptides between those lysine, it was after certain lysines and after certain other residues. And that is going to break the long chain of polypeptide chain into smaller fragments that are easily identifiable. And so you break them up. We then extract them back out of the gel through several pipetting steps, a lot of pipetting involved. We purify them, more pipetting. That's why I got my, wonderful carpal tunnel at the end of the day. But I did a lot of it, like day and night. And then we run it on, let's see if it goes to the next one. We ran them on a mass spectrometer. So we ran, we ran two different ways of identifying proteins, but we ran them on a mass spec machine where each individual amino acid was ionized and stripped off that chain and identified. So we sequenced every single one of those peptides, ran them into a database to identify them. We also looked at it through another method, which was called ICAD at that time. It's called different now, but it's isotope coded affinity tag. So all our peptides were tagged, and we then again ran them through mass spectrometer, and we looked for proteins in specific categories. And so doing that, we were able to find signature proteins that were only present in high-grade tumors or low-grade tumors, or that were overlapping in all types of brain tumors. And that's what we did to identify kind of a profile, a fingerprint for each one of the tumors. And that's what this one is showing. We had, and the number in front of it, that tells you how many samples we had for each grade. So this 45, 128, 107. So this is how we found attracted, right? So that was one of the spots. This is showing you for another spot. We then ran the mass spec. Each one of these peaks is corresponding to one of these amino acids. And then we sequenced it and used that to figure out what protein we were looking at. And we looked for whether that protein fragment was matching the molecular weight observed in our gels. So now going back to our actual attractant paper. So this is, by the way, what it would have looked like, just the different grades showing different profiles, different fingerprint. This is your control. This is grade 2, 3, and then 4. And then the primary brain tumors and metastatic brain tumors. So you see that they are very similar, but then there are certain differences that allow us to differentiate between them and tell when it is glioblastoma versus just a metastatic tumor from another place. So going back to our paper, which I had opened somewhere else. Where did it go? I'll open it again. from my downloads, so it looks nicer. No. No, that's it. It's not showing the way it should. Oh, there it is. Okay, so let's look at this one. So this was our first step. This is what we had talked about. And then we had talked about how when we looked at the Western blot, which is again looking at the protein, this time we are only looking for attractant, right? We used an antibody against attractant. And we looked for its abundance, and we saw that it was way more abundant in grade 4. What does this tell you? This bottom band, the transthyretin. Why do we have that one?
It shows its abundance everywhere.
Well, transthyretin is present everywhere. Why do you think I have it there? If it's present everywhere, why do I want to show it? Yep, exactly, Maya. It is a control. Anytime you have a Western blot, you will always see one protein that's a housekeeping protein that shouldn't change just simply because of the condition. That should remain consistent. And that's the internal control for us to show you that we loaded equal amounts of protein, or we loaded similar amounts of protein in all the samples. And that's what this one shows, that overall the number, the amount was very similar, but attractant was definitely upregulated in the glioblastoma samples. Good. So then we said, okay, we want to see if where this attractant is coming from, because attractant is present in the blood. And many times in high-grade brain tumors, you have breakdown of that blood-brain barrier. So maybe it's just coming from the blood. It has nothing to do with the tumor itself. So to look for that, originally I did tissue samples, right? I scrunched up the brains, I broke them up, I put them, extracted the protein, I loaded them, and I looked for attractant. And I did find that majority of my attractant, my high-grade brain tumors did have attractant upregulated, a lot more attractant there. But then my advisor said that doesn't prove that it came from the tumor. Why? And this is stuff that we had talked about last time a little bit. So
Because like you crushed everything.
and what else was there in the tissue other than the brain tumor cells?
Blood.
There were blood vessels. What else? Other types of cells? Yeah. It had the basement membrane. It had other types of cells. They weren't all astrocytes, right? It's going to have glial cells. It was going to have Schwann cells. It's going to have all types of cells. Who knows what else is there? So we can't really say just by looking at that it is coming from the tumor itself. So then to look at that, again, some more, we did what we called immunohistochemistry, where again, we use the antibody against the protein we are interested in, which is attractant. And this time we stained tissue sections. And these were paraffin embedded sections that were, saved from these brain tumors for a long time. And we took those sections and we stained them for attractant. And we looked at which area stained. So the top shows you just the tissue by itself, stained with, similar to what you guys did in class, in lab, where you stain them with the eosin, and then the methylene blue, this is hematoxylin and eosin. So the blues are the nuclei, the pink is the cytoplasmic space and the basement membrane, all that good stuff. And this is normal brain. And you see, there are very few cells there. It's very nice and clean. This is low-grade astrocytoma. You see a lot more cells now because they're dividing, replicating and dividing. And then this is high-grade grade 4 astrocytoma. And you see that not only are there a lot of cells here, but they're also now getting, disorganized. It's more and more disorganized. And there's a blood vessel here that's leaky. This blood vessel doesn't look like normal blood vessel. It's highly messed up. Normal blood vessels, they look like this track over here. So let me make it even larger so you can see, this is a normal blood vessel. This is a normal blood vessel, this little hole. They're very nice and clean and organized. Look what the blood vessel looks like right here. You even see all the red blood cells hanging out in there. This one also is a blood vessel, by the way, that, wanting to happen, but it's made, it's disorganized. And that's normal in tumors, that those blood vessels that are formed are highly disorganized. And that's what you see. And then this is another type of brain tumor called Schwannoma. Just to show an alternative control to show that attractant was specific for these astrocytomas and not for other types of brain tumors. And then we look at this. And this, actually, the normal tissue is what's important to look at. Why? Why is normal the most important slide to look at? So these are adjacent sections. So one section and right underneath. And I looked at exactly the same portion in the images, right? So it's the same site on the tumor. Why is the normal the most important? What does it show?
To compare it with the tumor cells, that shows no attractant.
Well, look at it carefully and say, is there any, so brown staining means attractant. Is there any brown staining in the normal tissue?
No. There is like some, just a few.
There's some. Where is it?
In E. Oh,
No. But where is it? Is it in the blue portion or the pink portion? Majority of the brown.
Pink portion.
Is it? So look right here, for example, at this arrow. This is an astrocyte. And right here, you see how it's blue? Not blue, it's brown.
Mm-hmm.
Here again, here again. Yes, it looks like attractant is in the cells. Good job, Ryan. So the cells are brown. What does that tell you? That attractant is coming from where? Is it coming from blood or is it? Yes, that attractant may be something that's normally found in brain cells, but not secreted, obviously, and found at some low levels right there. What happens when you look at a grade 2 tumor, astrocytoma? Now, where is attractant?
Outside the cells this time.
It's both inside and outside the cell. It's getting secreted and it is in certain cells as well. And the cells that are brown are a lot more brown now. They're not this faint little bit tiny brown every now and then. They are browner, right? They're a lot more brown. That tells you that one, see this actually, the normal tissue tells you that the most likely place that attractant is coming from is the astrocytes, because they do produce it in some level right there. And this shows you again that there is a lot of this brown here, attractant here, no blood vessel in sight, so it can't be necessarily from a blood vessel because it's right there without the blood vessel. And then look at the high grade tumor. What's important about this panel? Several things, but tell me two important things that we can take home from here.
All the attractant is secreted outside the cells now?
Not all. There's still pretty brown cells as well, but there's a lot more attractant. Good. The darkness of attractant blobs is very dark, right? What else? What about the blood vessel?
There is no attractant in the blood vessels.
What is it?
There is no attractant in the blood vessels.
Yes. Look at the blood vessel. It's clear. Those cells are not showing attractant. That means the attractant that originated in the CSF came from the tumor tissue and not from the blood vessels. And that was important. And then finally, when you compare to the Schwannoma tissue, which is very different looking, it does not have attractant. So obviously that wasn't because of another brain tumor, just the tumor. It's more specific to astrocytoma. Okay, cool. Now we get to why. So great, it's there, it's secreted, but why is it secreted? So to look for that, we started to basically work through one by one the various hallmarks of cancer. So if something is getting secreted by the tumor cells, it's getting secreted for a reason at the end of the day in most cases. And we wanted to know why. What function does attractant do that it will do this, that it will be secreted by the tumor cells? What advantage is it giving the tumor cells? So one hallmark of cancer, which is true for glioblastoma or high grade, really metastatic tumor like the one that we were looking at, is that they have the ability to migrate. They are very invasive. Astrocytomas are extremely invasive. They can just spread like wildfire. So we said, well, maybe attractant helps with that. The name certainly looks like it helps with that. Now, it's called attractant for a very different reason, by the way. It's an immune response protein. That's how it was originally found. So what we did here are two different assays, and I'll explain how the assays are done, and then you will tell me what they mean, what the figures mean. And both of these are meant to look at ability of cells to move and migrate. The first one is what we call a scratch wound assay. In this case, we allow the cells to grow fully on the plate. They completely fill up the plate. And then we manually make a scratch down the middle so that we can, create a blank space. We measure that scratch at the beginning. Once we make it, we measure it using our microscopes and measuring it, the actual width of the scratch at 3 different points along the plate. We then let them incubate in the presence of this antibody, not the antibody, but the protein attractant. We found some recombinant attractant, just the attractant artificially produced, and we dumped it in known quantities in the media. And we looked at how quickly those gaps were filled up over a 24-hour period. So we had our sample, which had no attractant. And then we had some at like 50 nanograms per mil, 150 nanograms per mil, all the way to 500 nanograms per mil. And we looked at them using 3 different glioblastoma cell lines. So these are from 3 different patients, 3 different genetic backgrounds. And we grew these cells. We first looked at the cells, we grew the cells, we scratched them, and then we added the antibody. What does that show us? Right there. So because this is taking away all the complexity even of CSF, because CSF has a lot of other proteins too. We just have media with artificially produced attractant protein added to the media in known quantities. So we know how much attractant is there, and there's nothing else there to confound the data. And we are looking at their cells' ability to fill up the gap, to migrate and to divide in that space and fill up that space over a 24-hour period. What do you find?
They found the cells with U87 has the highest Like ability to fill the gap.
Hmm. The greater the concentration of attractant, the faster the gap closed back up. Good. So panel A, that's all that it's showing. We don't know which cell, I don't know, I'm sure it says in there which cell line is showing in that panel, the first panel. It's one of the cell lines. So up here, it tells you that, the untreated cells, they filled up the gap about, 100 micrometers. So it's not like they didn't. They were going at it as well. They were moving, but they were still not fully closed up. Look at even 150 nanograms per milliliter. They're almost completely gone. The gap is almost gone in 24 hours, right? And then we used, again, our measuring techniques and our overall changes in the cells over our replicates to figure out the total distance migrated by the cells in each one of these conditions. What do we find in this panel when we quantified these differences? Does it work for all the glioblastoma cell lines or some of them or what?
I think it looks like it mostly looks like it works for all of them, but there is like a weird data point for the second bar on the
Oh, the
LN.
second bar, yeah, so I'm going to talk about the second bar. The first one is 0. That's 0 hours, right? Or 0 nanograms per mil. This second bar is actually BSA. It's another protein, non-specific protein that shouldn't have an effect on migration. So the second one is another control essentially. And in this case, I'm saying, well, what if I put 500 nanograms per mil of a different protein? Not attractant, something else. Will it still close the gap or not? And in this case, it looks like it doesn't do anything, right? In this one, it looks like it inhibited it. But for the other two, it didn't do anything. Basically, it didn't affect the closing of the gap issue, right? So the effect that you're seeing is real. That's what it tells you right there. The effect that you see from attractant is specific to attractant in this recombinant map. Well, that's great. Boss said, you're good. That shows that's promising, but that doesn't mean that attractant in CSF will have the same effect, because attractant in CSF has a lot of other things in there, and those other things may be competing with it and inhibit it, and maybe it won't have this function at all. So Ryan, yes, the U87MG traveled the most because it's the most aggressive. It actually moves a lot anyway. So in general, it just, if you look at even the lowest concentration or even no concentration, they tend to move a lot more. So yes, they are a little bit more susceptible to it though. So this time, actually, no, we didn't tell you that yet. So he said, okay, that's promising. Well, let's look at their ability to invade, because they don't just have to migrate, they don't just have to move, they have to actually invade through the cell matrix. They have to go through all that tissue and then be able to move. They can't just move. It's not an open field for them inside the brain. So that's the first question. So we did a second assay. This is called a Boyden chamber assay. In this case, we have a little tiny, it looks like a little sieve, a little basket, with a little sieve at the bottom. And we grow the cells on, it has a little coating on top that mimics the extracellular matrix. We grow the cells on the top part of that basket, and we immerse it into media where there is attractant on the bottom and the cells on the top. And we look at whether the cells chew through the matrix, get to the other side of the membrane. And again, this time we're still using the recombinant purified attractant, no CSF. What do you find? And this time, yes, the U87MG again is going to work a lot better either way, just because it is very aggressive. cancer. It has great ability to chew and do things that other cells may not have the same way. So it responds better. It does respond better to attractant. But what do you find? So what you're looking at here in these images are cells that actually made it to the bottom side of the membrane, not the top. So it would be if you took your cover slips and flipped them to the opposite side and looked at what went through to the opposite side of the plate.
The higher the concentration, the higher the migration was.
Yep. So in there as well, I see a couple of people responding in the chat. The greater the concentration of attractant, the more cells will invade and chew through the ECM. Good. And with greater concentration, the cell invades to a point. Yes. So you see that more and more cells are able to get through when there is attractant there. The higher the concentration of attractant, the more they move. And again, you kind of see that happening. But you also see there's a plateau effect eventually, right? So what can you, based on your knowledge of how molecule signaling pathways work, what would be causing that plateau effect? And we'll actually learn some of that today with our enzyme kinetics, but based on your current knowledge, you should still be able to think of that. Why would you see this plateau effect at some point?
Like when certain enzymes are all used up, isn't there like like
Yes,
it's a
enzyme wise, yes, once basically everyone has a binding partner, you can't fix any more response, right? So it's not necessarily an enzyme in this case, it's a receptor that the attractant is binding to maybe, or going through the signaling pathway with. Once everything is covered, once all the receptors are already responding, it's not going to continue to elicit that response. There's also maybe something, if you guys think about it, if you remember back to when we were talking about signaling pathways, we talked about positive and negative feedback loops. So there's also a negative feedback loop that can stop this from continuing to get activated. That's usually the case with most growth factors, that when they bind, they cause a reception and then it stops it. Right? So once it's fully activated, it will go back and stop further activation. So that kind of shows you that plateau effect, indicating that there will be some point where it can't happen anymore. So this is where they said, okay, well, great, but maybe it wasn't the cells migrating. Maybe the attractant was making the cells grow more. So maybe you are wrong in thinking that the cells are migrating. Maybe they're just dividing. They're going crazy. So we looked at their growth with different amount of attractant, all the way up to 4,000 nanogram per milliliter of attractant. What do we find? Over a 24-hour period, did they grow? Or 48-hour period this time? Did they grow?
No, kind of the same.
They overall remain the same. They didn't die and they didn't grow. So attractant definitely didn't kill them. It didn't really help them grow any more than they were already going to grow. So in this case, it's a relative number. So we are comparing each time point to its own control at that time point. So you're looking at whether it grew more or less than the control grew. And in all cases, it pretty much, remains very similar, a little bit higher in some, but not much. So that tells you that, well, they all grew and they should have grown, but they didn't grow in response to attractant is what it is. That's why the line stays pretty much around that same point, because they all kind of grew as much as they were going to grow. It never took off into a logarithmic change compared to the control. Cool. So regardless of the concentration, whether it was 0 or 4,000, they all pretty much grew about 50% compared to their 0-hour time point. So then definitely now going through our different hallmarks of cancer. The first one is unlimited growth potential. Does attractant give it unlimited growth potential?
No.
Does attractant give it, increased or decreased apoptotic ability or death ability?
No.
No, right? We know both of those because we don't see any evidence of either one in here. Does attractant increase their ability to migrate or metastasize?
Yes.
Yes. So that's how we kind of went through and looked at that. At this point, the idea was, well, great, it does it when it's purified. Does it still do it in the CSF? Can actual CSF make them do this? Same response. So we repeated the experiments, and this time we did either a controlled CSF, we did grade 2, grade 3, or grade 4 CSF. Now, if you look, there are 2 columns in each cell line. So this is again looking at migration ability, right, using our assays. And we are looking at migration abilities. These are in 2 different cell lines, LNC 308 and U87 308 MGD. We'll talk about this side of the equation in a second. Let's just look at the CSF. So you have control, grade 2, grade 3, and grade 4. They all have 2 bars, right? Why? What's with the 2 bars? What's the difference between the black and the white bar? No, they're not the replicates. Each one is composed of 3 to 6 replicates, however many times we did it. Probably 6 replicates, but
one has attractant and the other one, no?
No. They all have, so this is CSF. This is CSF. So whether the CSF had attractant or not is the question, nothing else. So we are taking CSF from patients that did not have tumor, that had low-grade brain tumor, medium grade, or high grade. That's what you have. So we assume, obviously, that these grade 4, grade 3 are going to have higher levels of attractant. Cool? What's the difference? What does this say over here?
What's the alpha and R?
Alpha anti, yeah. So this is your anti-attractant antibody. So one of these bars, the white bar, is showing CSF only added to the media on top of the cells. And the second bar shows media with CSF and anti-attractant antibody, or CSF that had been pre-incubated rather, with anti-attractant antibody. So we had the CSF, we either put it straight on there, or we added some anti-attractant antibody, left it in there for an hour, and then put that CSF on there. Why? These are some great questions for our finals. Why would we put? Maybe we are doing just this paper today. I like this. It may. How does it help indicate the presence of attractant?
By binding to the cell surface, if it was like inside or outside the cells. In
We're not
serum.
looking at them visually. We wouldn't know if attractant is bound anywhere. But if I incubate my CSF and with the antibody first, before I put the CSF on the cells, what happens to the attractant that was in the CSF? Where does it go? Where does it bind?
To the antibodies.
To the antibody. If it's bound to the antibody, is it going to bind to the attractant receptor?
No.
No. So will it work when it goes on the cells then? If it doesn't bind, will it work? Shouldn't.
No.
Exactly, it shouldn't work. So that's a control to see if attractant is the one causing the response. That's number one control. Now let's look at all these bars on the side. The first bar has no recombinant attractant, no antibody, no BSA. What is this? What type of a control is this?
Blank.
Okay. Other people should also talk, guys. There are just a handful of you talking. So yes, what is that? It's blank. What do you mean by blank?
Just normal CSF.
It's a negative control. There's no CSF.
No, okay.
This is just cells growing the way they grow. No attractant, no anti-attractant antibody, no BSA, nothing. Just cells and media growing the way they do and looking at what happens if cells have nothing on there. Do they migrate? Yes. Leslie shaking her head yes, and that's correct. Yes, they still migrate because they're cancer cells and they're invasive cancer cells. They migrate. So you have this basal level of migration that you will see regardless. Second one is in the presence of BSA. What is that? What?
Would it be a protein, another protein to compare?
Exactly. It's just like before, it's just a non-specific protein control to see, make sure that other proteins don't cause the same effect. Great. The third one, you have the cells incubated with the recombinant attractant. What is that? What would you consider this? So this was our negative control. Remember the very first bar. What's this?
The positive control, the one that has the attractant.
Exactly. That's our positive control. Right? Because that we know that should work because we've seen it work before, and that's what we are testing to begin with. Attractant by itself causes migration ability to be higher. And it is. It's twice as high. Great. It works. The next one is cells incubated with just the antibody. Why? What does that tell you?
If a still cell migrate without adding the attractant.
Yes, if the antibody itself can cause a response, because antibody will bind to maybe the attractant receptor or something as well. And does that cause the cell to get, the response and migrate? Does it? No. And then you have recombinant attractant incubated with the anti-attractant antibody added to the cell. What type of a control is that?
Like, doesn't it function as a negative control?
This is another type. Well, it's not a true negative control, but this is to make sure that it will act like a negative control. So this is kind of proof of concept because we are saying that if I incubate my CSF with the antibody before I put it on the cells, then it's going to not have the same effect, right? That's what I'm saying right here. So I want to first show that that's true. That's showing that. This is a proof of concept to show them that if attractant is bound to the antibody, it can no longer perform its normal function of increasing migration. So that's what this bar is showing, okay? And then we have our samples. What do they show? What do this, first of all, looking at the white bars only, what do you see happen in response to CSF from increasing grades of brain tumor?
Increases the migration of cells.
Yes. More and more cells are able to migrate when they are incubated with CSF from higher grades of tumor. The higher the grade, the more cells migrate. Is it all due to attractant?
Yes.
It's largely due to attractant, not all due to attractant, because if you look at the basal, it's still higher up here. You still see some addition. So yes, a lot of it is through attractant, but not all. Because when you add the antibody, you don't see a complete inhibition back to control, but rather some inhibition, a lot of it. Majority of the activity is due to that. And then looking at the second cell line, let's look at the second cell line. Again, you see similar response that, this one actually even more dramatic, but that's what you see, that attractant is able to migrate a lot more cells. And then in the presence of the antibody, that is inhibited greatly. So what does that show? So that's why even with those cringe-worthy Westons that we saw before from the tumor tissue, it still got accepted because of this figure, right? Because this was a very well thought out and planned out figure. It showed very clearly the response because of all the controls we did. And also shows the importance of developing a good experimental design.
It proves more about, it proves more that the attractant is the cause for cell migration.
Yes, of this highly invasive tumor, that it is a major cause of this movement. And so then, so attractant works with them to promote cancer cell. It doesn't grow the cells, it helps it move. It migrates them. It helps them move in their space as well as chew through ECM and migrate. So this one, the top only shows them moving, that it helps them move more. And then the bottom one right here, right, you can see this is your Boyden chamber, the ECM that they're chewing through, getting through. And again, you see that in the presence of controlled CSF, you have very few cells that migrate. The same number of cells migrate after incubation with anti-attractant antibody. In grade 4 CSF, pretty much like the whole membrane was covered in cells. A lot of cells were migrating to the opposite side. And when you compared it with the anti-attractant antibody, you look at this right here. There's still more cells in the control. There's still more cells migrating, but this greatly inhibited compared to CSF from the A4 alone. Yes, it's not going, we already looked earlier and we saw that it didn't help them grow any better than just normal. Like they grow the way they grow. It's not helping them grow any better. Okay, so that is this paper that.
So do you think this paper helps out with the treatment? They might like inject anti-attractant and at least inhibit the migration?
So it is something that they could work on, right? So this paper was published back in the day when I was doing my PhD. This is from 2006, I want to say, or 2005 even. No, 2006. So this was like right after I finished my PhD. In the meantime, it has made attractant as one of the biomarkers. So it was later on developed as a biomarker for high-grade brain tumors. So we do see that in the clinics, that they look for attractant to be there. The issue with developing it as a treatment would be that it would, again, it would be an invasive treatment. You have to find a way to get into the brain, to bind to the attractant, to work its way through. It's not as simple as it would be just taking a pill because, again, of the blood-brain barrier. And I am unaware of any clinical trials with it. I do know of studies that have looked at it and have found promising results, but I am not aware of anything that has developed it further than that at this point. Having said that, I also had a paper during that time with an anti-cancer molecule that I found that didn't even get published until recently and is only just beginning to get developed into a clinical model. So it takes a long time from bench to clinic. So high levels of attractant, in our case, all the studies we did, we actually did a institute trial after this looking at attractant, and we found that it was very specific to astrocytoma. So it is not the location, but the type of tumor. So these glioblastoma, astrocytomas that are known for their aggressive nature, for their really quick, basically, death of the person within a year, those are the ones that it is most associated with. Okay. Okay, I'm gonna stop this share for a second.
But like, I love how how you scientists think. Like, this is not enough. What we need to do to prove more, to like confirm that.
I will tell you that what you needed at that time is peanuts compared to what you need now to publish. So at that time, we could get away with just having this much and get it published. Nowadays, you need a lot more proof and a lot more types of proof, and before things can get published.
Oh, wow.
So I will, on that note, show you this thing, and then you'll see the difference in the amount of data that is required to publish. Close. 1 second, I'm sharing the screen again. Research. No, not this one. You can still publish, but if you want to publish in, like, really good fields or really good journals, you need a whole lot more. Where is the full text? So let me